Okay, let's talk about resting potential. So we know this as membrane potential. We've talked about this when we were calculating delta G values for molecules moving across the membrane. So you know that the resting potential of a cell is negative, around negative 60 to negative 70 millivolts. It's negative inside the cell because of large molecules like RNA and protein with negative charges, and the outside of the cell is more positive. So this normal membrane potential we are now going to refer to as resting potential, because you're going to see with neurons this potential or difference in charges across the membrane is going to change rapidly as signals are moved through the neuron. So neurons are special cells because they're electrically excitable. So they have these ion channels that we're going to talk about in a little bit, and they can change their membrane potential rapidly. So what maintains the resting potential in a cell? Well, by far the biggest influence is the potassium leak channels. So potassium leak channels, or any kind of leak channel, because we have some sodium leak channels over here, are channels that are continuously open. They're specific for the ion that passes through them, but the ion moves based on diffusion. So leak channels, always open, ions are moving based on diffusion. The reason that the potassium leak channels are so influential is because they are in the largest numbers. Right? So there's lots and lots and lots of potassium leak channels, fewer sodium leak channels. And don't forget our friend here, the sodium-potassium pump that helps maintain this enormous concentration gradient across the membrane. So if you look down in this chart, sodium and potassium have very big differences outside and inside the membrane. So obviously, sodium through the leak channels is going to come in, and potassium through the leak channels is going to go out based on diffusion, and our sodium-potassium pump helps maintain this difference in concentrations. So again, potassium leak channels, the largest influence on the resting or the membrane potential. Sodium leak channels are important too, potassium leak channels are important too, but the sodium-potassium pump is important, the sodium leak channels are important, but the main player is the potassium leak channels. Okay. So resting potential and action potential. So here we are, negative number, resting potential. This is where most cells stay. But again, with a neuron that can be electrically excited, what you have, and this is a graph of an action potential, is that you have an increase in the positive value inside the cell. So the membrane potential becomes positive. And this is called depolarization. So polar remembers opposite, so now we're going from negative to positive. At a peak here, and we'll talk about why it peaks out, the cell begins to repolarize, and it goes back to its more negative state. And we're going to talk about the channels that are important in regulating this. So in action potential, the membrane is going from negative to positive and back to negative. The other thing I want you to notice down here is hyperpolarization. So in fact, as the membrane is, as the membrane potential is becoming more negative, it actually overshoots the resting potential a little bit based on the ion channels that are opening and closing, and so you get a little bit too negative, and then the membrane comes back to its resting potential. We'll talk about threshold potential shortly, but let's talk about the ion channels that control the action potential. So these ion channels are called voltage-gated ion channels, which means they open and close based on change in voltage. So we've talked about ligand-gated ion channels that open and close based on a binding of a molecule. These are based on the charge across the membrane. So what it's trying to show you here is there's a voltage sensing region of that protein that when the voltage changes across the membrane, you get a conformational change and the channel opens. When it opens, the ions that it's specific for can now flow through, and when the what am I trying to say? When the membrane potential changes further, it goes back to closed, and we'll talk about inactivated in a minute. But again, these ion channels are based on changes in membrane potential. That's what triggers them to open and close. And this is what we call an all or nothing response. Okay? These channels are either opened or they're closed. They're not kind of open, they're not kind of closed, they're not letting some things through, it's either opened or closed. Right? So it's very distinct in the response you're going to get in the membrane. You're either going to get sodium ions flowing through, which are going to increase the membrane potential to the positive, or they're not going to go through. How do I get this one? Okay, so there's two main types, the sodium and the potassium voltage-gated ion channels. They look very similar. If you look at, there's two structures here, but they're made kind of different. So sodium ion channels are monomeric proteins. Mono means one. So if you look, this whole protein is all connected. All these transmembrane domains are all connected. And it folds into this channel, and right in the middle here, you can see a pore. On the other hand, the potassium are multimeric, and they're actually made of 4 individual proteins. So if you look carefully, there's a little space there. So these proteins, it's not one continuous protein like the sodium ones, but there are 4 the same. They come around and make this same kind of pore looking region. Another interesting bit about these gated ion channels, I told you they are specific. And your book talks about a selectivity filter. So within these 2 pores here, there are amino acids and oxygen lining them that interact with either the potassium or the sodium. Okay, and they have to interact in order to be able to go through that pore. So even though this channel is open, it is still very specific. And just another note that's important when we learn about action potential, sodium voltage-gated ion channels have a much more rapid response to changes in the membrane potential. Potassium acts a little slower. I don't know why that mouse doesn't work. Okay, and the last slide I want to talk to you about is this idea of inactivation. So these voltage-gated ion channels are not just open and closed, but they actually have an inactivated part of their cycle. So inactivated means they cannot open. So do you see you have to have this recovery period before you can get the channels opened. Okay? And we'll talk about why this is important for action potentials. Just so you know, both potassium and sodium channels are inactivated. The book shows you the sodium channel that has this what we call ball and chain. Inactivation particle. And so what happens is when it's inactivated, you get this part of the protein actually stuck up in that channel. Okay? And until that comes back out, the channel can't be open again. Potassium channels can be of either type, and your book doesn't go into this. I just want to let you know that potassium channels also go through a phase of inactivation. If they are of the C type, this is called the N type, they are inactivated up here at what we call the mouth of the channel, whereas you can see here this is on the intracellular side, this inactivation is on the extracellular side. And there's some interesting research trying to figure out if the inactivation here is due to constriction, so the protein components are coming close together and blocking, or some groups are proposing it's due to dilation, which means the space is too big for the potassium to come down. So, more information than you need to know, but I want you to understand that both types are inactivated, and the inactivated is kind of a beyond closed. You cannot reopen it until it recovers from inactivation. All right, we are going to stop there and come back talking about patch clamp technique.